Hello everyone, welcome to my YouTube channel Pharma Researcher. So this is a video number 6 in Telangana Pharmacist Grade-II exam preparation. The exam which is going to be held on the 30th of November 2024. So in this particular series, we are discussing total of 10 units. Each unit will be discussed along with their subtopics and the MCQs will be provided to you. And in this particular series, the total of 5 videos has already been covered, in which the first 2 units and the half part of the unit number 3 has already been covered. If anyone hasn't watched the remaining videos, so he or she can watch the remaining videos. I have provided the link in the description, or you can simply go to our YouTube channel Pharma Researcher. There you will find a playlist naming Telangana Pharmacist Grade-II exam preparation, and you will get each and every video there. And also for the PDFs, if you want the PDF of the question that were discussed in this particular video, so that PDF you can find the link in the description box. Simply click on the link, the PDF will open and you can simply download the PDF. And also I haven't mentioned the answer in the PDF, so in this coming weekend, so I will be adding the answers, and I'll update the PDFs for each and every video. So let's start with the video introduction. So this is the unit number 3, part number 2. Exam is on the 30th of November 2024. These are the different subtopics that are mentioned in this particular unit. So concept of health, concept of disease, this first topic, nutrition and health, demography and family planning, first aid. These 4 topics has already been covered in the previous video. Now in this particular video I am going to discuss these 2 topics, that is fundamental principles of microbiology and communicable diseases. So these 2 topics are very important. I have only included few MCQs, and I have given you the basic details regarding these topics, so that you can directly read from the PDF itself. And I'll be also explaining those important information within this particular video. So let's start with the first topic of today, that is fundamental principles of microbiology, microbes, isolation and staining techniques. These 3 are very important. Basically the different types of microbes, this is important, and the staining techniques, these are very important. And there are only few isolation methods. There can be a question, there may not be. So in the microbes part, I have included 3 main microbes, that is bacteria. Mostly you will see the question from bacteria only, or either you can find few questions from viruses or fungi. So only important questions I have included, because this is also a very detailed thing. So from the previous question papers of different state level exams and center level exams, so I have taken an idea like what type of questions can be asked from these topics. So let's start with the first question. So the question is, mode of nutrition of fungi is. So fungi, the mode of nutrition in fungi is heterotrophic. So here option C will be the correct answer. So there are different definitions like what is phototrophic, chemotrophic, heterotrophic, that you need to understand. So let's discuss one by one what is phototrophic, chemotrophic and heterotrophic. So in this, the photoautotropic means like these are those organisms which utilize the light energy. Those utilize the light energy from sunlight and several other elements such as carbon, or from inorganic compounds to produce the organic compounds like their food material. Then there are chemototrophic. Chemototrophic means these are those organisms that takes inorganic chemicals. These basically take inorganic chemicals and they transform these inorganic chemicals into the energy form. Then there are heterotrophs, that is a mode of nutrition in fungi. So these are those organisms, those get food from other organism, food from other organisms. So these heterotrophic organisms are also known as the consumers. Now moving to the next question. So the question is, mycology is the study of. So it is quite simple and direct. So mycology is the study of fungi. So here option B will be the correct answer. Next question is prions are, so what are prions? So prions are basically the it is basically a protein which is not having any nucleic acid. So only proteins without nucleic acid are prions. So here option B is correct. Then next question is the phase of bacterial growth during which the bacteria shows exponential growth curve is called. So there are different phases of bacterial growth. So basically, if you can see, I'll making a graph over here. So the bacteria will grow like this. Okay, or bacteria will grow like this. So basically, this one is the lag phase. Lag phase when the bacteria is adapting to the conditions or adapting with the environment, then there will be no growth. Then there will be a phase in which the bacteria adapted to that environment, and that shows it will show a rapid growth. This exponential growth is called as log phase. So here option B will be the correct answer. Then after some time this bacteria will be not showing any growth rate, not showing any decline rate. So that phase will be the stationary phase, when there is no change in the growth of bacteria. Then there will be a time when the bacteria starts declining, like the growth rate will be declining, more death rate will become higher as compared to new bacterial generation rate. So that will be the decline phase. So these are basically the four phases. I have included all the question on these four phases. You can check these questions. Then next question is the rate of generation of new bacterial cell becomes equal to the death rate during which phase? So when the generation, or we can say in very simple term, birth rate is equal to death rate in case of bacteria. So when these two rates are equal, then that phase is known as stationary phase that I have discussed. Then coming to the next question. So the question is during the lag phase. So what is mentioned in the lag phase? So I have explained you, like when the microorganism start settling to the new environment. Okay, so microorganism try to settle in the new environment where there is no growth of the microorganism, but it is adapting the different surrounding environment, surrounding conditions. So that phase is known as the lag phase. So here option A is the correct answer. Then next question is during the decline phase. So what happens in the decline phase? So in decline phase, the number of live cells decreases. Okay, so the deaths will increase and the number of live cells will decrease. So here option D is correct. Next question is cell of bacteria is made up of. So the bacterial cell is made up of peptidoglycans. So this is also an important question. So here option B is correct. Next question is the gap between cell wall and cell membrane is called. So the gap between cell wall and cell membrane, it is known as preplasmic or periplasmic space. So here option A is correct. So there is cell wall, there is cell membrane. So they have been separated by some gap. There will be a gap between these two. So this gap is known as preplasmic space. Next question is Micrococcus flavus is an example of. So basically Micrococcus flavus is a type of monococcus bacteria. So here option A will be the correct answer. If I'll talk about the example of Diplococcus bacteria, so it will be Diplococcus pneumonia. Diplococcus pneumonia. If I'll talk about the Streptococcus bacteria, then it will be Streptococcus pyrogens. If I'll talk about the Tetracoccus bacteria, so the example will be Gefkia, Gefkia tetragena. So these are the different examples of these different types of bacteria. Then next question is rod shaped bacteria are known as. This is very important. In different exams, the question has been arise from the shape of bacteria. So the rod shaped bacteria are known as vacillize. So here option C is correct. If I'll talk about the vibrio, vibrios are the coma shaped bacteria. These are the coma shaped bacteria. If I'll talk about spirilla, so spirilla are the spiral shaped bacteria or helical shaped bacteria. And if I'll talk about the cocci, cocci are the spherical shaped bacteria. These are the spherical shaped bacteria or ovoid shaped bacteria or ovoid shaped bacteria. So these are very important. Bacilli and cocci, these are the two most important, most repeated type of bacteria. Next question is bacteria which gain energy from sunlight and get electrons from inorganic compounds are known as. So those bacteria which take energy from the sunlight and get electrons from inorganic compounds, these type of bacteria are known as photolithotrophs. So here option B will be the correct answer. If I'll talk about the photoorganotrophs, so these are basically the simple organisms such as protozoas. They basically derive its energy from the photosynthesis. They derive energy from the photosynthesis and from the organic compound, not from the inorganic. They are taking energy from the organic compounds. Then talking about the chemoorganotrophs. So chemoorganotrophs are those species or those organisms that uses chemical bonds. These uses chemical bonds in organic compounds or in oxygen as their energy source and take electrons or hydrogen from organic compounds. So these are the chemoorganotrophs. Then there are chemolithotrophs. So these are the organisms which uses inorganic, these uses inorganic reduced compounds as a source of energy. So these are the different types of bacteria based on the source of energy. Next we can move to the another question. The question is gram negative bacteria have. So what is the difference between gram positive and gram negative bacteria? Basically here we can find. So there is a very important difference, like the gram positive bacteria have very thick, it has very thick peptidoglycan layer in the cell wall. What gram negative bacteria has? Thin peptidoglycan layer. So in case of staining technique, like if you have heard about the gram staining, so the in gram staining the gram positive bacteria have like very thick layer, so it will not capture the counter stain. That's why it will appear as violet in color. Meanwhile the gram negative have the thin layer. That's why with by using alcohol the primary stain will be dissolved and the counter stain will be captured. That's why it will show the pink color. So these are the basic difference and very important. Now moving to next question. So the question is which of the following bacteria is responsible for the spoilage of refrigerated food? So the bacteria which is responsible for the spoilage of refrigerated food is Listeria monocytogenes. So here option A is correct. Next question is microorganisms that can only survive under high hydrostatic pressure are called. So those microorganisms which can survive on high pressure are known as verophiles. So here option B is correct. Then next question is microbes which can survive in the presence of oxygen, what they do not use oxygen for its metabolism are called. So those microorganisms which survive in the presence of oxygen and they do not use oxygen for its metabolism, these type of microbes are known as aerotolerant. So here option C is correct. Next question is genetic material is transferred from one bacteria to another bacteria in bacterial conjugation with help of. So in order to transfer a genetic material in bacteria, there are different processes like the conjugation, transformation, transduction. These are the different methods for the transfer of genetic material in bacteria. So they are basically talking about the conjugation, like in bacterial conjugation, with the help of what part this genetic material is transferred. So this one is pilus. So here option C is correct. Next question is molecular cloning of recombinant DNA can be done with the help of. So the molecular cloning of recombinant DNA. So it can be done with the help of transformation process. So basically it is also a method of transfer of genetic material in bacteria. Next question is which viruses are involved in the bacterial transduction process? So in bacterial transduction process, bacteriophages are involved. So here option C is correct. So this part is very important. Like they're asking about virus, like which virus is involved in transduction process in order to transfer the genetic material. So this one is the bacteriophages. Next question is transfer of genetic material can take place in bacteria through which method? So this I have already explained. So there are different methods: transformation, transduction, conjugation. So here option D, all of the above is correct. So let's talk about the conjugation. What is conjugation? So in this particular method, the genetic material is transferred from one bacteria to another in contact, like when they were in contact with each other, then the genetic material will be transferred. Then there is transduction. This we have discussed, like there will be a transfer of genetic material from recipient to donor with the help of bacteriophage bacteriophages. Then there is a transformation process. So it is the process of DNA uptake by the bacteria from the surrounding environment. Okay, from the surrounding environment, DNA or genetic information can be taken up by the bacteria. Then moving to next question. So the question is cell wall of fungi is made up of. So the cell wall of fungi is made up of chitin. So here option C is correct. Next question is mode of reproduction in fungi is. So the mode of reproduction in fungi is both sexual and asexual method. So fungi mostly reproduce through sexual and asexual methods. So here option C is correct. Next question is zygomycota are also known as. So zygomycota, these are basically the molds. These are also known as bread molds. So here option A is correct. Next question is no sexual stage is found in which class of fungi? So in Deuteromycota class of fungi, there will be no sexual stage. So here option C is correct. Basically in this particular type of fungi, the sexual reproduction in is absent. Then moving to next question. So the question is Deuteromycota class is also called as fungi imperfecti as. So it is also known as fungi imperfecti. Why? Because there is no sexual reproduction in Deuteromycota. So here option C is correct. Next question is viruses without nuclear envelope is called as. So those viruses which do not have nuclear envelope, these are known as naked viruses. So here option B will be the correct answer. Then moving to the next question. So the question is in viruses nucleic acid can be present in the form of. So in viruses the nucleic acid can be present in the form of both DNA and RNA virus. So here option C, both A and B is correct. Next question is leucoviruses have. So what are leucoviruses? So these are basically the viruses which have both DNA as well as RNA as their genetic material. So here we can say option C, both DNA and RNA as their genetic material is the correct answer. Next question is according to Baltimore classification, viruses have ssDNA as their genome can be put in which group number? So there is a classification of viruses based on Baltimore classification. So there are total of 7 groups as per the Baltimore classification. So the group 1 will give it is double stranded. It is double stranded DNA. Its characteristics are this. In group 2 it is single stranded DNA. You can say ssDNA, that is in the question. So option B will be the correct answer. Then for group 3, it is double stranded RNA. It is double stranded RNA. Then there is group number 4, which is having single stranded RNA positive. And in group number 5, there is single stranded RNA negative. And then there is group number 6. So group number 6 denotes the single stranded RNA viruses with reverse transcriptase. So I have added this table also at the end of this at the end of the MCQ. You can recheck and revise these particular classes. There is also given the mode of mRNA production and the several examples based on the each group, so that you can also study. Then one class is left, that is the class number 7 or group number 7. So it is double stranded DNA viruses with a reverse transcriptase. Okay, so these are quite simple. So if you want to remember, so first two are DNA based, the next four are RNA based, and the last is again DNA based. Then you can simply remember the double stranded and single stranded. So from the table itself you will be able to easily read and remind the things. The next question is, India ink method is used for. So it is a staining method, and India ink method is basically used for capsule staining. So here option B is correct. Next question is, teichoic acids are the main surface antigen of which organism? So this question is very important. This question has been asked in different exams. So teichoic acids are basically present in gram-positive bacteria. So this can be a point of difference, like in gram-positive bacteria teichoic acids are present, and in gram-negative bacteria teichoic acids are absent. So these are basically the surface antigens of gram-positive bacteria. So option B is correct. Then next question is, which of the following is not an acid or acid fast organism? We need to identify like which is not a acid fast organism. So mycobacteria, ascospore of certain yeast, spirochetes, exoskeleton of insects. So here spirochetes, it is not an acid fast organism. So here option C will be the correct answer. Then next question is, Fontana's method is for staining of which of the following? So Fontana's method, it is again a staining technique, and it is the staining technique for spirochetes. So here option B is correct. Then next question is, virus mediated transfer of host DNA from one cell to another cell is known as. So it is a virus mediated transfer of DNA. So virus mediated, we have discussed one question earlier, like virus mediated one is transduction. So here option A will be the correct answer. And which virus is involved? It is bacteriophages. Then next question is, bacteria spores are, so bacteria spores here, if you can see option, so allow the bacteria to multiple in adverse condition, are usually formed by gram negative bacteria, can be identified with gram stains, or are killed by temperature of 120 degrees Celsius for 20 minutes. So bacterial spores are basically killed, or the sterilization temperature is 120 degrees Celsius for 20 minutes. So this statement will be the correct for bacteria spores. Next question is, stains useful for identifying fungus include the following except. So which of the following stains are used to identify fungus? And asking for except. So basically which is not useful for identifying fungus. So first is gram stain. So gram stain is not used for the identification of fungus. It is for the bacteria. Usually to differentiate gram positive bacteria with the gram negative bacteria. So other names you need to remember in this question, like to for identifying of fungus, the stains used are hematoxylin and eosin, gomori methenamine silver, and periodic acid scaf. So these three names you need to remember in case of staining of fungus. Then we can move to the next question. So the question is, autotrophs is or are bacteria that. So autotrophs are those bacteria which synthesize all their organic food. Means they do not depend on some other organism. They synthesize all their food by itself. So they are the autotrophs. Next question is, Jale-Nielsen staining is done for. So Jale-Nielsen staining is a staining technique that is done for mycobacteria. So here option A is correct. Then spirochetes are also important. We have a question previously. Like for spirochetes, it is the Fontana's method. Fontana's method or Fontana staining is used. For eastern fungi, we have done a question like the three different types of stains which are used for identifying fungus are hematotoxin, hematoxylin, and eosin. Then gomori methenamine silver, and periodic acid scaf. So these are the different stains that are used for identification of fungus. Next question is, capsid is present in. So where the capsid is present? So it is basically present in the viruses. So here option B will be the correct answer. There can be some other questions regarding yeast. So yeast is basically comes under the group of fungi. So this question is also very important. So this point you can remember. Also regarding the viruses, so viruses in viruses there is a DNA that is enclosed in a protein coat. Okay, DNA is enclosed in a protein coat. Then moving to next question. So the question is, which of the following is an oncogenic virus? What is oncogenic virus? Oncogenic viruses are those viruses which can cause cancer. Okay, which can cause cancer. So there are basically two types of oncogenic viruses, or we can say oncovirus or oncogenic viruses. Either they can be DNA viruses or they can be RNA viruses. So the examples coming under DNA viruses and RNA viruses are important, because sometimes question can be asked like which of the following is DNA virus, which of the following is RNA virus. So these are important. First we can discuss regarding the DNA viruses. So DNA viruses include human papilloma virus, that is HPV. It is a human papilloma virus. Then there is HBV, that is hepatitis B virus. Hepatitis B virus, or I can say hepatitis virus, it is very important because hepatitis B virus comes under DNA viruses, and hepatitis C virus comes under RNA viruses. So here you can remember DNA is HPV. Then next is Epstein-Barr virus. It is very important. So Epstein-Barr virus. Then there is human cytomegalovirus. Then there is Kaposi's sarcoma associated herpes virus. Then there is Merkel cell virus, Merkel cell polyoma virus. So these are the different examples of DNA viruses. Then moving to the RNA viruses. RNA viruses. So RNA viruses include hepatitis C virus, that is HCV. Then there is human T lymphotropic, human T lymphotropic viruses. Then there is one Rous sarcoma virus. So these are the RNA viruses. So examples you have to remember. So if here you can see poliomyelitis virus, human papilloma virus, varicella zoster virus, or herpes simplex virus. We can say human papilloma virus, that is a HPV virus. It is a oncogenic virus which comes under the category of DNA viruses. Then moving to next question. So the question is, which of the following microorganisms is prokaryotic? So we need to identify like which of the following is a prokaryotic microorganism. So basically microorganisms are basically categorized into two broad categories. Either they are cellular type or they are the non-cellular type. Non-cellular type. So in non-cellular type, it will include viruses, viroids or prions, viruses, viroids or prions. And in cellular there are two categories. One is prokaryotic, other is eukaryotic. Now the prokaryotic includes all the bacterias, the algae, blue-green algae, while the eukaryotics include fungi, molds, protozoa. So these comes under eukaryotic. So here the question is simple, like which of the following is prokaryotic? So here you can say bacteria is a prokaryotic microorganism. So here option B will be the correct answer. Then next question is, which of the following are the methods of isolation of microorganisms? So there are different methods of isolation for microorganisms. For example, the streak plate method, the pore plate method, the serial dilution method. So here all of the above will be the correct answer. Apart from these three methods, there are more methods that are used for the isolation of microorganisms. For example, single cell isolation by micromanipulator. There is a direct transfer method. So if I'll talk about the streak plate method, so simply in streak plate method, what happens? So we take a small amount of sample and we have a petri dish. In this petri dish, there is a solid nutrient medium, which is sterile in nature. And in this, we'll add this small amount of sample, which is of microorganism, and we can make the streaks in this petri plate on the solid nutrient medium. And this sample is streaked by the nichrome wire loop. It is done by the nichrome wire loop. So with this help, it is streaking is done. So this is important, like which wire is or which loop is used to streak the sample in streak plate method. So it is the nichrome wire loop. So this is important. Now coming to second method, that is the pour plate method. So in pour plate method, simply the pouring is done, and in order to obtain the pure culture by isolation, why pour plate method, there we need to do the serial dilution. What is serial dilution? Serial dilution means we have different test tube. So with 5 I can say, I'll explain you. Like here we can add 1 ml of sample, 1 ml of sample, and the other 9 ml will be the water, distilled water. We'll mix that. So this is a primary dilution. Now we'll again we'll take 1 ml from this test tube number A. 1 ml will be added in the second test tube, and the remaining 9 ml will be filled with the distilled water. This will be the B dilution. Again, now we will take this 1 ml from test tube B, add in the test tube C, and the remaining will be filled with the water. Then we'll take 1 ml from test tube C, and we'll add in the test tube D, and remaining 9 ml will be filled with distilled water. So like this way we will be diluting the medium. Serial dilution is this. So basically the contents are basically uh, incubated, or plates are then incubated for specified time period to obtain isolated colonies. Basically after serial dilution we will be having a Petri plate. Here we will be having the nutrient medium, and from the final dilution we will be adding the sample. We will add the 1 ml sample to this nutrient medium, which is placed in the Petri plate, and there will cool that and solidify that, and the incubation will start in this way. Then if I'll talk about the serial dilution method, it is a similar type like serial dilution is done, and then some of the tubes of higher dilution showing growth will be found to contain only one colony of species, which can be observed by microscopic examination. Then if I'll talk about the other two methods like the single cell isolation. So in this the important thing is micro manipulator is used. Micro manipulator is used in combination with a microscope. Microscope is basically used to visualize the things. Then there is a direct transfer method. Direct transfer method. So sometimes bacteria, yeast and molds can be found in pure culture under natural conditions also. So from there they can be directly cultured and directly transferred to the suitable medium and incubated to get number of colonies. So this is the direct transfer method. Now moving to the next question. Next question is, in which method of isolation of microorganism micromanipulator is used? So this I have explained you, it is used in single cell isolation method. So here option C is correct. Next question is, which of the following method for streaking a solid culture medium? So method is used for streaking a solid culture medium. So in order for streaking, which is this streaking is done in the streak plate method. It is done in the streak plate method. So streaking can be done in a square method. Like in the square method, there can be 4 quadrant method, and it can be done in the zigzag manner. So these all are the different methods in order to streak the microorganism in a solid culture medium. So here option D is correct. So these are the few very important questions from this particular topic. Now I have added you few important points that are equally important, and the question can be framed from these points also. So the first point is regarding the gram staining. So in staining technique, gram staining is very important. So there are basically 4 steps in gram staining. It is basically used to identify the gram positive stain and gram negative stain. So what happens is applying a primary stain to a heat fixed smear of a bacterial culture. We'll take a bacterial culture and we will apply a primary stain which will give a crystal violet color to the bacteria. Then we'll add a trapping agent. Gram iodine is added. So after adding gram iodine, it will trap the color inside the like on the surface of the bacteria. Then we'll do the rapid decolorization with alcohol or acetone. So we'll simply use acetone or alcohol to decolorize this color. Then after decolorizing, we'll add the counter staining with safranin, and basic fuchsin is sometimes substituted for safranin counter stain. So in this, what you need to remember is the 4 step. First is initially the crystal violet color will be formed. Then the trapping agent is gram iodine, and for decolorizing alcohol or acetone is used. And for counter staining, safranin is used. So this question has been asked previously, so this is also important. Like for counter staining, safranin is used, and sometimes basic fuchsin is also substituted for safranin. Now how to identify the gram positive and gram negative bacteria? So what happens, the gram positive bacteria which is having a thick membrane. So due to thick membrane, this color will be there, and the trapping agent will trap this color, and there will be no decolorization with the alcohol. So it will not adapt the color of the safranin. Then it will show the blue or violet color only. Then there is a if there is gram negative bacteria, so what happens, so the membrane in gram negative bacteria is thin. So due to this thin membrane, this trapping agent and the decolorization is easily done with the alcohol, and the counter stain will give its color to the gram-negative bacteria. That's why it will appear red or pink, because they are counter stained. So these are important things. Basically here you need to remember the color most importantly. Gram positive, blue or violet color, mostly violet. Gram negative bacteria, it will be having red or pink color. Then next point is regarding the Gill-Nielsen stain. So this question we have already discussed, like which of the microorganism will have the Gill-Nielsen stain, that is the mycobacteria. So Gill-Nielsen stain positive will be with mycobacteria and nocardia, and it will be negative with lactobacillus, corynebacterium or actinomycetes. Then there are aerobic and anaerobic bacteria. So this can be asked in the exam, like which of the following bacteria is aerobic or which of the following is anaerobic. So here you can find like Bacillus anthracis and Bacillus subtilis. These are the aerobic. Aerobic means they use the oxygen. Anaerobic means without the presence of oxygen. So these are Clostridium tetani, Clostridium botulinum, then Clostridium perfringens. So these are the important key points. Now we can move to the next part, that is I have added a diagram for the classification of bacteria. So here you can find the classification with respect to each and every categories, like based on shape, based on gram staining, based on oxygen demand, based on pH of growth, based on mode of nutrition, based on osmotic pressure requirement, temperature requirement, number of flagella, spore formation. So this is a very broad classification. You have to learn these all the classification, like you can simply pick up any one of the category or any of the category. For example, you can pick the cocci. You can simply Google search like what is cocci. So they will give you like based on the shape, like what is the shape of cocci, so that you can mention. What is the shape of bacilli, that you can mention. So these I have already discussed in few of the question. Gram staining I have already covered. Aerobic and aerobic I have covered. Few questions from these this we have covered. So this the other are not that much important. You can remember these: obligate halophiles, facultative halophiles, based on the temperature requirement. So these two you can simply Google search the term and you will find the definition. Mostly that definition will be asked. Now we can move to the next part. So as I have told you in the question regarding the classification of viruses, there is a Baltimore classification. There are 7 classes, and here you will find the characteristics, like the first group is having double stranded DNA, and mode of mRNA production is basically mRNA is transcribed directly from the DNA template, and the example is herpes simplex. So here the most important thing is you need to remember the characteristics and you need to remember the examples. So this is the most important. So here you can find out like first two are DNA. Only two things you need to remember, like either it is double stranded or single stranded. So first two are DNA only. First one is double stranded DNA, second is single stranded DNA. Then third, fourth, you can find out like the third one is double stranded. Again, it will be RNA. Fourth will be single stranded RNA. But now in single stranded, there will be positive and negative terms. So fourth one is positive single stranded RNA. Fifth one is single stranded RNA negative. Then for the sixth and seventh, these are with the reverse transcriptase. So it will be a single stranded RNA virus with the reverse transcriptase. It is double stranded DNA virus with reverse transcriptase. And this, then rest you can find out the examples, like rabies is very important, herpes simplex is very important, hepatitis B virus and human immunodeficiency virus is important, and rotavirus. These all you can memorize these examples. Now we can move to the last topic of this particular unit, that is communicable diseases, causative agent, mode of action, prevention of chickenpox, measles, influenza, diphtheria, whooping cough, tuberculosis, and these all diseases. So in this particular section, I will be covering few of the questions, and I have provided you the list of these important things. So in this particular topic, causative agent is the most important. Definitely you will get the question from this particular topic. Maybe you can get the one question, or maybe you can get the two questions, because there are limited topics in this particular unit. So let's start with the first question. So the question is, which of the following is sexually transmitted disease? So the sexually transmitted disease among these are syphilis. So here option D will be the correct answer. If I'll talk about measles, so it is basically the respiratory infection. Typhoid is intestinal infection. Intestinal infection. Plague is arthropod borne infection. Then coming to the next question. So the question is, which of the following is respiratory infection? So among these respiratory infection include tuberculosis, influenza, pertussis. These all are the respiratory infection. So here option D is correct. Next question is, typhoid is, so what is typhoid? So typhoid is basically an intestinal infection. So here option B will be the correct answer. So there are different types of infection and different types of categories will be there. So if I'll talk about the respiratory infection, so there are different diseases in respiratory infection. For example, chickenpox will be there. Chickenpox will be there. Measles is a respiratory infection. Influenza is a respiratory infection. Diphtheria is a respiratory infection. Whooping cough or pertussis and tuberculosis. These all are the respiratory infection. Then in intestinal infection, typhoid is there. Apart from typhoid, hepatitis is there. Hepatitis is there. Cholera is there. Then poliomyelitis is there. Food poisoning is also there in intestinal. Hookworm infection. So these all are the intestinal infections. Then coming to arthropod-borne infection. So it includes the plague, malaria, filaria. So these all are the arthropod-borne infections. Then coming to sexually transmitted diseases, these are very important. So here comes the AIDS, syphilis, gonorrhea. So these are the sexually transmitted diseases. Now moving to the next question. So the question is, rabies is a, so what kind of disease is a rabies? So rabies is simply a viral disease. So here option A will be the correct answer. Next question is, the diseases are transmitted from insects to human are, so those diseases which can be transmitted from insects to the humans. So these type of infections are known as vector-borne diseases. So here option C is correct. Then next question is, which of the following is a viral disease? So among these, the viral disease will be the influenza. So here option D will be the correct answer. Next question is, malaria is transmitted through. So malaria is a condition which is transmitted through female Anopheles mosquito. So here option A is correct. Next question is, TB is a dash disease. So TB, tuberculosis, it is a bacterial disease. So here option A is correct. Next question is, a disease that spread from one person to another person or animal is. So those diseases which can be spreaded from one person to another person, these are known as the communicable diseases. So here option D is correct. So all those diseases which are mentioned in the syllabus, those are the communicable diseases only. So apart from those, for example, diabetes, cancer, hypertension, these are the non-communicable diseases. So in this way you can learn. Next question is, World Health Organization recommended a control strategy for tuberculosis known as. So the WHO has recommended a control strategy for tuberculosis, that is DOTS therapy, which is directly observed treatment short course. Directly observed treatment short course. So full form can be asked, so you can remember the full form as well. Next question is causative agent of chickenpox is. So the causative agent of chickenpox is varicella zoster virus. So here option A will be the correct answer. Apart from this, if I'll talk about the RNA paroxysmal virus, measles, then Corynebacterium diphtheria is for diphtheria, and Bordetella pertussis is for whooping cough. Whooping cough for pertussis. Then next question is causative agent of tuberculosis is. So the causative agent of tuberculosis is Mycobacterium tuberculosis as well as Mycobacterium bovidae. So here option C, both A and B is correct. And this Vibrio cholera, it is a causative agent of cholera. Next question is the causative agent of typhoid is. So the causative agent of typhoid is Salmonella typhi. So here option A will be the correct answer. If I'll talk about the Clostridium botulinum, so it can be the causative agent of food poisoning or botulism or botulism. Then moving to the next question. So the question is hookworm infection is caused by. So the hookworm infection is caused by Ancylostoma duodenate or Necator americans. So these both are the causative agent for hookworm infection. So here option D, both A and B is correct. And Yersinia pestis, it is the causative agent for plague. Next question is Treponema pallidum is causative organism of. So Treponema pallidum, it is a causative organism for syphilis. So here option A will be the correct answer. If I'll talk about gonorrhea, so for gonorrhea it is Neisseria gonorrhea, it is the causative agent, and for AIDS it is the HIV, human immunodeficiency virus. And if I'll talk about the rabies, so for rabies the causative organism is Lyssa virus type 1. Then moving to next question. So the question is causative agent of measles is. So the causative agent of measles is RNA paramyxovirus. So here option D is correct. Next question is poliovirus in oral polio vaccine causes production of. So oral polio vaccine in oral polio or poliovirus in oral polio vaccine causes the production of immunoglobulin IgA. So here option D is correct. Next question is which of the following is waterborne disease? So the waterborne disease among these are amoebiasis. So here option A will be the correct answer. Then next question is food poisoning mainly caused by. So food poisoning is mainly caused by Salmonella species, E. coli, noroviruses. So here option D, all of the above will be the correct answer. Next question is DPT immunization agents for. So DPT is the immunization vaccine for diphtheria, pertussis, and tetanus. So these can be immunized from DPT vaccine. So here only diphtheria is mentioned. So option D is correct. Next question is which is a surface infection? So which of the following is a surface infection? So it is the leprosy. Leprosy comes under the surface infection. So option B is the correct answer. Next question is which is intestinal agent? So among these the intestinal agent is cholera. So here option D is correct. Next question is incubation period of diphtheria is. So the incubation period for diphtheria is 2 to 5 days. So here option A is correct. So in different books you will find the incubation period as different values, but not much change, but there can be a slight change. Next question is the incubation period of gonorrhea is. So the incubation period for gonorrhea is 2 to 5 days only. So here option D is correct. So now with this particular question, we are done with the MCQs. Mostly I have covered all the causative organisms here. But apart from this, I have provided you few of the information, text information that will be useful you to revise all the things. So I have added it in the end only. So you can revise these things once you will get the PDF. So there are common communicable diseases include these different categories: respiratory, intestinal, arthropod, sexually transmitted, surface antigen, and rest. In these you can find like which one is viral, which one is causing by bacteria, which is causing by any other agent, so that you can read with the causative organism. So respiratory infections include chickenpox, measles, influenza, diphtheria, tuberculosis, whooping cough. Intestinal include hepatitis, cholera, typhoid, poliomyelitis, food poisoning, hookworm infection. Arthropod 1 include malaria, filariae, and plague. Sexually transmitted diseases include AIDS, syphilis, gonorrhea. Surface infection includes rabies, trachoma, leprosy, and tetanus. So these are the important categories. These categories you need to remember. Next, I am going to discuss with you the this particular table, which is showing you the diseases, incubation period, causative organism, mode of transmission, and prevention. So in these, I will be explaining few things, and I will be mentioning you what is most important, so that you can remember mostly. So basically, first diseases, I have included all the diseases in this particular table that are given in the syllabus. So for chickenpox incubation period, you can remember causative organism is the most important thing in this table. Rest important thing, mode of transmission is quite easy, because mode of transmission will be almost similar in most of the diseases. Like it will be a droplet infection, so through sputum or through sneezing, few droplets will infect the person, or with the contamination from direct personal contact. Indirect contact means by using the infected towels, by using the used blades of the infected person. So these are the common mode of transmission. So here you can find few, if any specific is there, that you need to remember. For prevention, mostly there will be a uh, immunization programs, like the vaccination will be there, so that are important. If vaccination is there, that is important. If any drug is there, that is most important. So that I'll remember. Mostly causative organism is for sure the most important thing. Now we can move to the next is measles, that is also known as rubeola virus. So it is caused by the RNA paroxysmal virus of rubella virus. Direct personal contact or droplet infection, this is quite simple. And measles vaccine will be given, so there is no specific name, so nothing to worry. Influenza is influenza virus AVC, so this is quite simple. Here you can remember the drug, like the amantadine drug, so this is important. Then for diphtheria, corynebacterium diphtheria, causative organism is quite clear with the name itself. Then droplet infection, direct indirect contact. Then in diphtheria you can give the DPT vaccine. For diphtheria, pertussis or whooping cough, and tetanus, in these you can give the DPT vaccine. Next, with the pertussis, again Vortadella pertussis, and several cough preparations, antibiotics, and also the DPT vaccine. Then for tuberculosis, Mycobacterium tuberculosis and bovidae. And here you can prevention can be done with the sputum detection test are there. Isoniazid can be given, paraminosalicylate acid, ethambutol. BCG vaccine is the most important thing that you have to remember. We have discussed this question in the previous subjects also. So BCG is Bacillus calmette-guerin, so this is important. Next is poliomyelitis, so it can be caused by the poliovirus, single stranded DNA RNA virus. Then it can be prevented by oral polio vaccine. that is the Sabin, and there is a Salk polio vaccine, which is the parental, which is given by the parental route. Then there is hepatitis A, hepatitis B. Simply hepatitis A virus will cause this, hepatitis B virus will cause the hepatitis B. So these can also cause cancer. These are the oncogenic viruses. So not hepatitis A, hepatitis B is a oncogenic virus. So that we have discussed in the question. So these can be transmitted through sexual contact also. This can be transmitted through mosquitoes, and here you can give the human normal immunoglobulin vaccine, or here you can give the hepatitis B immunoglobulin vaccine. Next is cholera, caused by Vibrio cholera. It can be transmitted through general things like the contaminated water, food, milk. It can be transmitted through flies, direct or indirect contact. ORS therapy can be given immediately after the diagnosis. ORS therapy can be given, and personal hygiene is the most important thing. And cholera vaccine, definitely that will be given for the prevention. Then coming to typhoid, Salmonella typhi is the causative organism, and TAV vaccine, that is typhoid, paratyphoid A, paratyphoid B, is the combination of TAV vaccine, and chloramphenicol is the drug of choice for typhoid. This is important. Next is food poisoning. So I have discussed one question like in which I have mentioned many different organisms which can cause food poisoning. In that, the important one is Clostridium botulinum. Other organisms you can read from the question itself. So it can be ingestion of different foods which are contaminated with either living bacteria or their toxins or chemical substances. So through that, food poisoning can be developed. So here I have provided the mode of transmission, like from which chemicals, from which plant and animal sources, or from which bacteria it can be transmitted. And in this, we can simply use preventive measures like properly cooked food we should eat. We should store the surplus food in the refrigerators, and other preventive measures we can take. The next is hookworm infection. Here the Encyclostoma duodenal and Nectar americans, these are the causative organisms. And for the incubation period, you can simply check it on the Google, because the variations are a lot in the different books and different sources. So this, you can also skip this part. So in this, the female worm eggs in feces can transmit the hookworm infection. In this only we can use the hygienic washrooms, and personal hygiene is the most important thing in this. Then there is a plague which can be caused by Yersinia pseudotuberculosis or Yersinia pectis. So there is a vector of plague which is a rat flea, and it can be transmitted between the rodents by fleas. And the destruction of rats, fleas by insecticidal sprays. So these are the different preventive measures. Next is the malaria, which can be caused by different Plasmodium vivax, Falciparum, malariae, ovale. These are the different causative organisms, and it is transmitted by the female Anopheles mosquito. So preventive measures can be, we should avoid water storage in open area. We can use chloroquine or quinine in case of malaria, and mosquito repellents can be used to be protected from the mosquito bite. Then there is filariasis condition, which is a mosquito-borne disease. Causative organism is Wuchereria, Vencrofti, and Brugia malai. And there are two types of mosquito that can cause filariasis, which is Vencroft's filariasis and Malayan filariasis. And in this, the most important thing is DEC, that is a diethyl carbamazine. It is the drug of choice in case of filariasis. This question is asked multiple times, so this is very important. Also, we can protect ourselves from mosquito bites, and similar preventive precautions we can take as we take in the malaria. Next is rabies. Rabies is also known as hydrophobia. Another name you can always remember. It can caused by Lyssa virus type 1. Dog white saliva of rabid animals. So mainly through saliva or dog bite. It can spread from person to person, and there is no cure of this particular disease. If a person has this particular disease, rabies, then it is 100% death causing disease. We can have a preventive measures like anti-rabies vaccine can be given, but if the disease has spread in the person, it has developed in the person, then there is no cure. So preventive measures will be the isolation in dark room and the cleaning of wounds and scratches with soap and water as early as possible. So these are the preventive measures. Next is trachoma, which is a eye disease. So this question is also asked like, what is trachoma? So trachoma is related to eye. So it is a eye disease caused by chlamydia trachomatis, and it can be spread direct or indirect contact with ocular discharge of infected persons through flies, swimming pools, contaminated water. Preventive measures are anti-fly measures and personal hygiene only. Then there are several other diseases like tetanus, which can be caused by endotoxin of Clostridium tetani. So it is contaminated through wounds with tetanus spores, injuries caused by iron articles. Preventive measures can be DPT vaccine you can give. You can also give the long-acting penicillin. For leprosy, which is also known as Hansen's disease, this is important. This name you need to remember. Caused by Mycobacterium leprae. Name is very easy. So this question cannot be asked. Maybe in order to make the question paper difficult. So these type of direct will not be asked. Then it can spread through breast milk, droplet infection, direct or indirect contact. Depsone is the drug of choice for leprosy. This drug is very important. Apart from this, rifampicin, clofazimine, isolation, these are the different preventive measures and treatment measures. Next diseases are syphilis. So syphilis is caused by Treponema pallidum. Most important question. This question has been asked in multiple times. In each and every question paper, you can see this question. Then it can be spread through sexual contact. It is a sexually transmitted disease, and it can spread from mother to child as well. So preventive measures can be avoiding sex with multiple partners or by the use of condoms during intercourse. So these are the preventive measures. So all the STDs will be having the same preventive measures. Then there is gonorrhea, which is also sexually transmitted disease. It is caused by Neisseria gonorrhea. Same mode of transmission like sexual contact, indirect contact through infected maternal use. Same preventive measures. And again for AIDS, it is also a sexually transmitted disease. It is caused by human immunodeficiency virus, that is HIV, and it is spread through sexual contact through blood or transfusion. Mother to child, it can be spread, and again the same preventive measures. So this is a complete table. Most of the things, important things have been covered. You can cross check the incubation period from other sources if you have any specific book regarding microbiology, so you can check from that. Rest the causative organism in this is most important. And with this particular video, we will be completing the unit number 3. And next we will just start the unit number 4, which is regarding the pharmaceutical chemistry. So that's all for this video. We will meet in the next video. Till then, thank you very much.